try and find a seat. There's a few more up on the balcony as well, if anyone wants to go up there. The TAs are walking, for those of you in Bio 3200 anyway, the TAs are walking along the aisles, and if you forgot to bring a piece of paper, because we're going old school with notes with a piece of paper, or even if you're not from Bio 3200, you just want a note to take some notes, just put your hands up and they'll get to you with paper. They also have golf pencils that are pre-sharpened, and so they'll get to you too. So Resh, you see hands up over here. And Miriam, if you want to come into the middle, there's some hands over. If you want to come around the middle side, there's some hands over here. Up on the balcony, there's 2 TAs as well, so just put your hands up if you want some paper. Miriam, there's 1, 2, 3, 4, 5 people over here. Okay, I know it's really, really full today, so. And again, if you're not Bio 3200, you are most welcome to paper and pencils as well. Just don't hand them in. Alright, so for the people just coming in now, if you want paper and a pencil, especially if you're Bio 3200, and if not, put your hands up and the TAs will come get to you. Anyone? Resh or Miriam over here? Okay, and for those of you who are in Bio 3200, before we forget right now, we're going to put our name and student number over there, guys. Yeah, name and student number on that piece of paper right now. If you're not Bio 3200, if you're cardiovascular center, you don't have to. Okay? At the end of Dr. Howlett's talk, those of you who are Bio 3200, we have a very organized way that we're going to do to pass all the papers over to the center and over to a row up at the top so that we can pick up the papers and everybody doesn't have to rush to the front. Okay, we ask everybody to stay for the entire talk, and if for some reason you can't or you have to run out to the washroom or something, you're welcome to hand your paper to your neighbor if you trust them to hand it in for you. Okay, anyone else need paper and pen? Okay. All right, so before we start, I'm going to tell you very briefly who we are. And so usually we do this as a cardiovascular center specific event with graduate students and faculty, but we're in the cardiovascular section of our course today, and we have the great honor of being able to bring a very special speaker to you today. Probably Kevin can't hear me. Okay, so I'm going to tell you very briefly about the Cardiovascular Center because it's relevant not just to the faculty and graduate students, but the undergraduate students as well. So we basically hail from all across campus, so from the University of Guelph. So our faculty are in the Ontario Veterinary College, College of Biological Sciences, Biomedical Sciences, Clinical Studies, Population Medicine, Molecular and Cell Biology, Human Health and Nutritional Sciences, Integrative Biology, Animal Biology, Engineering, and others. And we have a website that you can look on to see who the faculty members are. So basically, we're a Senate-approved center. It was approved by the University of Guelph Senate in 2015. And so our goal is to transform our thinking about cardiovascular disease, which is our leading cause of death in the world, to combine our forces from basic research to clinical translation. So we have everybody from molecular biologists to biochemists to people doing human translational medicine and people doing animal medicine. And basically our goal is to discover new ways to diagnose heart disease, advance therapies, and train the next generation of clinicians and scientists, which is you guys. So we do have a website, and I encourage you to take a look. If you Google CCVI, you'll end up at the high school down the road, which is our acronym, unfortunately. But if you Google Cardiovascular Research Guelph, you'll end up at our website. So over the years, we have distinguished scientists come and visit us, and the purpose of that is threefold. The first is that we like to bring in experts who are doing cutting-edge research internationally so that we can see what's going on around the world. The second is we like to be able to profile our research and network our graduate students and faculty to those world leaders. And the third is we like to network people, ideas, and resources, so that would include everyone in this room having opportunities to look at, what's out there and make the connections. And I know that many of you are attending, if you were free, the networking events later on. And we've had all sorts of great success, including publications in some of the highest impact nature journals and other places that have resulted from our meetings. It's also increased our funding, so funding is really important if you want to go to grad school, and I think we've been awarded $14 million in competitive research funding in the last 5 years. As undergraduates, you wouldn't know, but from all the major research funding agencies in Canada. We also run student executive things, and this is to build the CVs of the students. And so we have one that is for postdoctoral and graduate students, which tends to represent each of the members' labs. And we also have an undergraduate executive, so we can start to plan forward of what the needs of undergraduate students are. At the moment, I have one undergraduate student executive, and I can certainly take on a couple more, and you're welcome to email me if you think that's of interest. It's not a lot of time. It's just sort of centers around when we do events. Okay, so that's a little bit about that. And now I'm going to tell you about our distinguished cardiovascular scientist, Dr. Susan Howlett. So Dr. Howlett did her bachelor's degree in biology and psychology at Concordia University in Montreal. She then did her master's degree at the Memorial University of Newfoundland in St. John's, Newfoundland, and her PhD as well, followed by postdoctoral studies at the University of Alberta, Edmonton, and now is a professor of pharmacology and medicine, and especially geriatric medicine, at Dalhousie University. So very briefly, she is an internationally renowned scientist and has led discovery research into how age, frailty, and sex steroid hormones affect cardiovascular function and predispose towards heart disease later in life. So what you're doing now and how that affects aging and the aging of your parents and grandparents. She has been continuously funded from the highest levels of funding in Canada, so CIHR or its predecessor, the Medical Research Council of Canada, or MRC, and has held over $6 million in operating funds. She's an associate editor of the Journal of Gerontology, associate editor for aging research, for mechanisms of aging and development, and has guest edited issues of Current Opinion in Physiology, Mechanisms of Aging and Development. She's also co-chair of the Dalhousie Cardiac Excellence Research Wave, called Dal Crew. So for anybody who wants to get into graduate work in research, she is the head of the unit there at Dalhousie University. And she has extensive leadership experience, which she's been very humble on this piece that I'm reading here, but she just says experience at local, national, and international levels, but she's got her hand in pretty much everything in the cardiovascular world. She has published 186 peer-reviewed papers, 12 book encyclopedia chapters, and has given more than 131 invited national and international presentations. She's participated in the European Mouse Age Initiative, the Canadian Frailty Network's workshop on frailty biomarkers, and was invited to Parliament Hill to present frailty work to the Parliamentary Health Research Caucus and was the only Canadian invited to the NIH US Working Group on Sex Bias in Cardiovascular Research. Her personal and collaborative experience allows insight into the needs of researchers across the career span, and her mentorship of now generations of young scientists, the great bulk of whom have pursued academic careers, speaks to her ability to inspire young researchers. Okay, so what we do, and you may or may not have seen, those of you who are in the undergraduate cohort, is we're going to invite our speaker up now, and we're going to present her, come on up, Dr. Howlett, with a plaque, so she'll remember all of us and the time she came as our keynote speaker to the University of Guelph. And a small honorarium because we're so pleased to have her here. So we're going to give her just a minute to figure out how to put all these back on, and then I'm going to turn this over. At the end of the lecture, you're welcome to ask questions, or you can save them if you're in one of the meeting groups. Okay, so that one is Kevin.
Okay.
And it's being filmed, just so you know. Up in the corner, there's Kevin.
Hi, Kevin. Do you have liftoff?
We do.
Good. This is the one that's really live, right?
Yes.
I blew into earlier and destroyed your drums. Yeah, sorry about that. Okay. Thank you. Can everyone hear me? Okay. Yeah, I just don't want to. How about now? Up top, we got. Oh, great. Okay, thank you. Thank you, Tammy, for the generous introduction. I appreciate it. And for the opportunity to come and talk to everyone here today. This is a big audience, and it's going to be fun to talk about the work that I'm doing. And if I lose you from time to time, I hope to kind of regroup. And if you have questions, please don't worry about asking them at the end, or if some of you are coming to, I know we're going to meet many people later on. I'm happy to talk to it. Don't worry about, silly questions. There's none. I want to try and communicate some of the, the joy that I find in what I do. And career in science is really fun, and we often don't emphasize that, talk about grants and things, but I've had a lot of fun, and I've been a scientist for a long time, so happy to talk about any of those aspects with you when we meet later on. But right now I'm going to talk about the research that I've been doing on mice, and talk about mouse models that better reflect the people who actually get cardiovascular diseases, and why age and sex and frailty, a concept you might have heard the word but may not know much about, how these things can impact on cardiovascular diseases and why they're important to think about. I do tweet. I used to tweet more often before it became X. Science Twitter is fun anyway, so I have enjoyed that in the past and may get there again. So this was the situation certainly when I first started off in science, and that was many decades ago. And most biomedical research is done on male animals. That's a public health concern. You can find this in the Northeastern University news article of 2019. And so, that's really interesting. And it was such a problem that this prompted the National Institutes of Health in 2014 to publish this article called The NIH to Balance Sex in Cell and Animal Studies. And that's because people were using one sex in a lot of their studies, and most often it was male. And so the National Institutes of Health, who give money to scientists all over the United States, and lots of it, to do research, were saying, unless you have a particular reason for only using one sex in your studies, you need to use both, because we need to have work that we can translate to a broader swath of the population. So this caused a lot of people to actually shift gears, maybe not as much as you might have thought. So this is a study that was published in eLife in 2020. And what you can see, I don't have the pointer, but in any event, I think what you can see here is in the blue in 2009, these are studies done in males only. And you can see that by 2019, these are studies in all disciplines of biomedical science, that actually the number of studies done in males only has decreased, which is good news, I think. And the number of studies done in females is in red. The number of studies using both sexes has definitely increased, and that's good to see. But I would say that the group that bothered me the most is the unspecified group. So this is where you read a paper and you look at the methods section, and they don't bother to say whether the animals they used were male or female, or the cells that they used were from male or female animals. So this is actually a huge problem for the reproducibility of science, right? Because you can't even go back and reproduce the experiment because you don't really understand what was done. So I was really happy to see that that bar is decreasing as well. And I would also add that there are some reasons why you might want to use just male animals if you were doing work on, for example, prostate cancer, some studies with testosterone, or if you were doing work in pregnancy, for example, in females, you might have a really good reason to do this. But still, I think we still have work to do, because there's still a huge number of studies that are only using one sex, and in many cases, people will give excuses like it's too expensive. But the granting agencies in Canada as well have been pretty clear that we need to try and up our game a little bit here. So I'm kind of proud of this. This is the title of the very first paper I ever published in Science, and it's called Sex Differences in the Phenotypic Expression of Avian Dystrophy. And it turns out that chickens were a model of muscular dystrophy. They were kind of bred to have really large breast muscle, lots of white meat, and it turned out that they ended up developing a strain of chicken that had muscular dystrophy. And I went to work on this project. Started out working in skeletal muscle as a graduate student. And I think the reason that I paid attention to sex differences, when we were using adult chickens, and they were New Hampshire Reds. And I mean, if you look at them and you think about their muscles, the males are almost twice the size as the females. And it occurred to me that this might be an important thing to look at. So we did look at a lot of the muscle properties, and we found that the females, interestingly, were affected more severely early on, but the males became aggressively much more severely affected. So there were differences, even though the gene for this is not X-linked, or sex-linked. And so this was an interesting, I thought this was really what one should do, but it turns out that it was not considered to be all that interesting until more recently. And why is this important? Because I said I was talking about heart disease, and I have worked for my entire career, postdoctoral and independent career, on the heart and heart disease. If you look here, these are data from the American population. Canadian data are very similar. And what you can see is that this is heart disease in men and women as they're increasing with age. And you can see that men and women, there's some differences in the percent of the population who have heart disease. And here you can see that women below the age of menopause are less severely affected. But by the time they get to be 80, in fact, women have slightly higher incidence. And the diseases that men and women get can differ as they get older. And I think the thing you can say when you look at this slide is that your risk of getting cardiovascular disease varies across the life course. It may be lower at some ages in women than men, but clearly by the time you get to be over the age of 60, there's a high chance that you'll have some aspect of cardiovascular disease. It might be hypertension. This is all diseases, but the main risk factor for developing cardiovascular disease is age. And so I was interested in the sex difference aspect and also the aging aspect here, and I thought we might like to try to look a little bit more closely at what happens in the heart as we age, and I've done a lot of work on that, and what happens in the heart that differs between males and females, and I've done a lot of work on that. And this is a project I'm going to describe now, where we looked at the ventricle. So this is, you're doing the cardiovascular unit now. This is the heart, this is the ventricle, the cells. This is a picture of a cell from my lab, and it's a ventricular myocyte, rod-shaped and striated. And it turns out these cells have receptors for sex steroid hormones, including estrogen, and androgen receptors. And the genomic receptors mediate the chronic effects of these hormones on the heart. But there's another receptor that is called a non-genomic G-protein coupled estrogen receptor, or GPER. And this receptor has been much more newly described, and it mediates the acute effects of estrogen. So when you put estrogen on, effects that happen very quickly. And so I'd like you to keep that in mind, because we're going to come to how we managed to use that to and develop a new approach clinically. So we've used in all of these studies I'm going to show young and old, or just old mice, C57 black 6 mice, which are kind of a laboratory strain that's very commonly used in different labs. And most studies are done on males and females. And so this is a study that I did with a student in my lab who came from Nepal, Anjali, and she did a master's degree in my lab, and she's now doing a PhD in Australia. So one nice thing about science careers is you get to travel. Anyway, Anjali and I worked on this project. So this is a complicated system, but some of you may have seen it in labs. It's called a modified Langendorf perfused heart. And so basically we have a heart that has a balloon in it that can measure pressure, and we can perfuse it through the aorta, and so we perfuse the circulation, and we can keep the heart alive for quite a long time with a solution inside that mimics normal blood, if you will. And we can measure the beating, and then we can look at it afterwards. We can expose it to low oxygen called ischemia, and we can look at it. We developed a way to do what they do in cardiac surgery, which is they use a solution called a cardioplegic solution. And this is a solution that keeps the heart from beating, has a lot of potassium in it. And they put it in an ice bath. They keep it really cold, so it's like 4 degrees Celsius. And they will put this solution into the heart for a short period of time. These are the cardiac surgeons. And then they will arrest the heart or stop it from beating. Then they can do surgery on the heart because it's not moving, right? And so we were interested in this, and I had worked with some cardiac surgeons at Dalhousie to try and develop and tweak some of these solutions. But we thought maybe we can try and harness some of the benefits of sex steroid hormones like estrogen in this system. And we're able to use this cardioplegic solution and put it into the heart for 6 minutes, and then we just stop all the blood, all the flow. Okay, so then the heart sits there with no perfusion at all, just like it would during cardiac surgery with this protective solution inside. And what we then did is we did experiments measuring pressure. I'm not going to go into that, but at the end of the experiment, we took the heart down and we cut through it, like you can see here with the dotted line. And we can get a cross section where you can see the left ventricle, which is the big round part, and then the right ventricle, which is more squashed, if you will. And we stain it with a stain called PTC, or triphenyltetrazolium chloride. And with this stain, the viable tissue is red, and the dead tissue is kind of pink or white, right? So you can see that this heart has quite a lot of what we call infarcted tissue, or not viable tissue, dead tissue, cells that didn't make it, when we expose the heart to this ischemia, or no flow. And so here's what we found when we did this experiment on hearts from young and old male and female animals. So I've shown the examples here, as you can see, but if you look at the mean data, the infarct area, or the damaged area, is higher in males than in females, but it's especially very low in the old males, which was kind of surprising to us. So the largest infarcted area, which is almost half the heart, was seen in the young males, but the older males had smaller infarcts. The females, it didn't seem like age made so much difference. They had smaller infarcts anyway. So we thought about what could be going on, and we thought, well, maybe testosterone is playing a role. So we measured testosterone, and we saw that it was greatly reduced in the males. We didn't see much testosterone in females regardless of age, but age reduces testosterone in male mice. And so that was interesting. So what we then did was we used a mouse model where they're chronically exposed to low testosterone. So it's something called a gonadectomy model, but they have low circulating testosterone, which we verified. And we looked at the infarct area, and we found the gonadectomized mice had very small infarcts. So it suggests that in this model, testosterone is actually having a detrimental effect on the heart and its ability to survive, and that this was really interesting, and we were able to publish it. And so we thought, this is quite an interesting finding. One other caveat I should mention is that the older males also had a lot of fat tissue. And fat tissue has an enzyme in it that can convert whatever testosterone's around into estrogen. And so they also had higher levels of estrogen when we measured it. So that could be also having some beneficial effect here as well. So we wondered if some of the beneficial effects of estrogen, the females always survive these types of insults better than males, regardless of the age, and we've done other studies on this. So we wondered, could we actually harness some of the beneficial effects of estrogen to improve these solutions? This is a neat idea, I think, because this is something that you can translate, means like transfer to people quite easily, because these cardioplegic solutions are in use in the operating room. We already did one study where there's a solution that we changed, and they actually were able to use it in kids, and we actually drove it for them in some of the animal studies. So we wanted to add a GPER agonist, because it has to be something that acts fast, not something that acts long-term, which would change gene expression. So we added a GPER agonist, which is something that mimics estrogen at this receptor. And we added it to the cardioplegia solution in the older hearts. And what we found was pretty encouraging. So we see here that we've been able to take the infarct sizes. These are all from old animals. We take the infarct sizes from here about 30% in the females and about 20% in the males, and reduce them to 10% or less just by adding the G1 to this solution. So it looks like you can actually kind of start to tweak these solutions to get a better outcome in people with cardiovascular disease. And the other thing that's relevant about this is that a lot of the people who are going to be going for such surgeries on their heart will be older. So it's really interesting. If we were doing these experiments in younger animals, we would have come up with potentially quite different conclusions. And so finding out that the older male hearts actually appear already to resist this type of injury, and still finding new ways to improve it, is a really exciting type of experiment to do. And it can lend itself to translation, to changing practice in the clinic. So while we did these experiments, I'm going to do a bit of a pivot here. While we did these experiments, we looked at the mice. And as you might have heard at the beginning, I did an undergraduate degree in what's now called neuroscience. Biology and psychology was what it was called at the time. And so I actually started out doing work in the CNS as an undergraduate. My honors thesis was on that. Then I moved to skeletal muscle, and then as a postdoc I moved to the heart. So, science allows you to move around a bit, so don't feel too shoehorned into, well, I only do this one thing. I've certainly, reinvented myself a lot of different times doing different experiments because I kind of got curious about something. And so back around 2010, I got curious about looking at these older animals we were working on back then. And I saw that some of the aged animals, these are all C57 black 6 mice, they're supposed to be genetically very similar. They're often litter mates. We bring them up, they were 2 years old. An old mouse, about 75 years old, is about a 2-year-old mouse. Bring them up to the lab, and some of them are, hanging by one arm from the bars, beating their chest and feeling, 2-year-old mouse. And then we got a 1.5-year-old mouse who really looks quite hiding in the corner and not very happy. So it's like, okay, some of the animals look like they have age-related deficits. Some of the others look very, robust and springy. And this is, not unlike maybe what we see with some older people. You'll have older people in your lives, and this is a sign from the United Kingdom, which I think is terrible. Elderly people, and the internet's had a lot of fun with it. People have tried to kick this version out. So this isn't how people really want to age, right? They want to age more like the middle cartoon. And in fact, people do age at different rates, right? We know this. I take inspiration from people like Ed Whitlock, one of the world's greatest marathon runners. Here he's seen at 85 years old. Ed runs a 3-hour and 56-minute marathon at Toronto Scotiabank Marathon. Like, that is pretty awesome time for people half his age or less, right? This is Olga Ketelka. She's setting a world high jump record. This is a woman who's doing the high jump over the age of 95. Like, people fall, you know, when they're younger than her and break things. She's doing a high jump, right? This is Fauja Singh, who now lives in the United Kingdom, originally from the Punjab. He does an 8-hour and 25-minute marathon at 100, but he actually improved his time, you know? These are quite amazing feats, right? And so people do age at different rates, and there are examples of really successful aging that you can see here. Some people are fit at any given age, and some people are frail. So the idea of frailty, it's a word that we use kind of colloquially just in conversation. And it comes from the actuarial literature. So it comes from demographers who are doing, I don't know, kind of trying to figure out for insurance companies who's going to live longer and who won't. And they talked about frailty to describe the idea that some people who are 65 years old, you know, have a much higher risk of an adverse health outcome, like substitute death, heart attack, whatever, than other people of the same age. And clinical medicine has been interested in this idea of frailty because it conforms with what they see in the hospital, which is people of the same age with, you know, wildly different outcomes from the same procedure. And the ones that don't do well tend to start off frail. So how do you measure frailty? Because you can't just look at someone and tell. If you just look at Ed Whitlock's face, you know, you'd say, oh, there's an old guy, he's probably frail. No, actually, you know, he could kick your butt in the marathon, right? So you can't just look at someone and tell, right? So clinicians have come up with some different scales to try to measure frailty. The one developed at Dalhousie University is called a frailty index, and it's based on the accumulation of health deficits. The idea is that on average, the older you are, the more things you have wrong with you. But some people have many fewer things wrong with them at a given age than others. So the frailty index is a really simple concept. The frailty index is simple math. You get a list of deficits. Say you have 50 things on the list that could be wrong with you, everything from fallen arches to diabetes. And you say, do they have this or not? So you count deficit, yes or no, 0 or 1. If you have 50 deficits, someone has 25 of them, they have 25 divided by 50, which is a frailty score of 0.5. So the frailty index can vary between 0 and 1. In practice, it never gets as high as 1. We can talk about that later. So this was first put forward in 2001, and this tool is now used all over the world. It's part of the United Kingdom's electronic health record. So there's now millions of people who've had this index calculated for them in their primary care physicians. Interestingly, even though the idea was developed in Canada, in Nova Scotia in particular, it is not on the electronic health records yet here, but it is in the United Kingdom and other places. So what did they do? So I know you can't see this. This is called a comprehensive geriatric assessment from Capital Health in Halifax. I'm hoping you can see this. So what they do is they take a patient to measure frailty, and what are these deficits, right? So they have this form, and they fill this form out. This form has about 70 deficits when you get it filled out, so a lot. But some of the things shown here are, if you see nutrition, how's their nutrition? Good, fair. So you can see, if you look over to the far side of the screen, it's poor. Elimination, they're incontinent. They're dependent on ADLs, which are activities of daily living, like feeding, bathing, dressing. In people, you can ask, okay, what were they like 2 weeks ago? And if 2 weeks ago they were continent, they were independent, you can tell something went wrong with this person. This person suddenly got a whole lot frailer. Maybe they've got pneumonia, maybe they've got, but they suddenly got a whole lot frailer. And so this is the kind of approach that is used clinically to measure frailty scores. And in terms of sex differences, because sex matters with frailty too, there's this interesting thing, which is called a morbidity-mortality paradox. The idea is that at any given age, this is going from 57 to 87, women are frailer than men, but they live longer. So are they frailer or are they more robust? It's hard to know, right? But in any event, they have higher frailty scores. And this is found pretty well by everyone. And there's a million explanations for it. Some of them have to do with gender. Some of them have to do with immune system. People are really not sure the reason for this, but it's a really interesting and fairly reproducible finding. So, okay, frailty. I was talking about mice. I saw mice look like they were aging variably, like people. So I wondered, are mice frail? So I first wondered about this with a student, Randy Parks, in my lab in 2012. And I'm just going to say a little bit about this. This is the first attempt, because I thought, oh, I'm not a veterinarian. There are veterinarians here, but I'm not a veterinarian. I didn't know how to assess frailty in mice. And I didn't know how to do a little mini comprehensive geriatric assessment on mice. So I guess I can't put my hand up or something funny happens, okay? I'll stop gesturing. So I thought, okay, I can measure data. So, scientists measure data. So I measured activity levels. So we had an open field, so we looked at how much they moved and where they moved and how much they reared, et cetera. I measured their body composition. So I measured bone mineral density, percent fat, et cetera. I measured hemodynamic parameters. This is a tail cuff. You can actually, a little sphygmomanometer you can put around the tail of a mouse and get the blood pressure. It doesn't hurt them. And I measured blood work. I got a small blood sample and we were able to do routine blood work. From these types of data, I was able to generate a list of more than 30 items that I could measure. And I got okay, here's the average in the young animals, and then here are the scores that deviate from that. And that's how I did the very first frailty index. Published in 2012, had to send that paper to 3 different journals before I got it published. It is now a citation classic, so don't give up. I didn't give up. I shopped it around a lot, but people kept saying, this is a very fascinating idea, but, and the but was really, oh, we've never heard about it before, we've never thought about it, and we don't want to publish it. But the Journal of Gerontology Biological Sciences took a chance on me, and I'm on their editorial board now, and as editor and stuff. I was very grateful for that. So this was a proof of concept. The reason I'm talking about this briefly is because we took the hemodynamic idea, that blood pressure and heart rate and things, and the idea that routine blood work could give you some interesting data, and we translated that back to people. So this is in 2014. What we did was we did something called an FI lab. And that is, we looked at blood work from a bunch of people, and we saw what's normal potassium. Well, the low cutoff is 3.8 millimolar, the high is 5. And so if you were below or above serum potassium, you got a score of 1 for that item. If you were in the normal range, you got a 0. And if you were too high in your serum sodium, you got a 1. So we took every single solitary thing we measured, like urea, glucose, you name it. We added them all up, we divided by the total number, and we got an FI lab score. So you can even get a frailty score when you're not looking at people, but measuring a lot of data from them. There's some differences. You can pool all the data together. We've published this, I worked on it with Joanna Blodgett and Ken Rockwood. We've published it, and there's a big review published in Age and Aging just in 2023. It predicts mortality. It's a really interesting tool. And so anybody sitting here with access to clinical data from people who has this type of data, I could certainly help you or talk to you about how to create a frailty index. And it adds information about overall health, about your population, right? So I thought, okay, the paper with all those different measurements worked, but that was a ton of work. It took a long time. Those are not fast. A lot of those techniques are not fast. And I thought, can we actually do what they do in clinical medicine? Can we do a little comprehensive geriatric assessment on mice? And so with Jocelyn Whitehead, who was at the time an honors student just finishing up, worked in my lab on this project with me, and we looked at a ton of aging mice and young mice. And we talked to the veterinarians, and in fact, our veterinarian at our university is one of the co-authors on the paper. And we looked at all the things that can happen to mice as they age, and looked at things that were generally wrong, would have a bad consequence. And so we looked at things like tumors, distended abdomen, curvature of the spine, all kinds of things. And again, we thought if they were absent, they got a score of 0. If there was a small evidence of this, it got a 0.5. And if it was large, it got a 1. Again, adding them all up, dividing by the total number of things we measured by, we got a frailty score. I'm just keeping an eye on the time here. And this is a really interesting, this is 14 scores from 14 mice and, I don't know, 10,000 plus humans. And we plotted them, we normalized the age as a percent of the maximum lifespan, and these are the frailty index scores. I think what you can see is frailty index scores increase exponentially with age. You can notice that the highest frailty scores are only 0.5, right? And in fact, the highest frailty scores that are seen in people are about 0.67. So it's like when you have a ton of deficits, when you get about 2/3 of them, if you get one more deficit, you're not going to survive. It doesn't matter if you broke a finger, if you got a cold, like it could kill you when you're that frail. So it kind of explains why frail older people were so susceptible to things like viruses, for example. We've had lots of ample evidence of that in recent years. So we see that the data from the mice just overlay that curve in humans really nicely. So we think this is a fundamental kind of biological process that's going on, this deficit accumulation that starts to render the organism or the person frail. We published in Nature Aging in 2021 a review, if anyone is interested in this. This deals with everything from the clinical data on frailty to the mouse data to the physics of frailty, which is a lot of mathematical modeling that I've also engaged with, and Ken Rockwood has also worked on that as well. So what about male-female differences? We saw that women were frailer than men. And so these are the clinical deficits. You can see here, there's 17 months on panel A and 23 months on panel B, and these are all the different deficits they can get. And these are the clinical frailty index deficits. And you can see in some cases the males, which are in the white bars, are frailer, have more deficits than the females. And in other cases, the females have more deficits than the males. It's hard to know looking at all the data, really, which is frailer. But when we calculate a frailty score and we look at the average data, you can see that even in the mice, the males are less frail than the females at different ages. So this was a large data set. We needed large numbers to see this difference. We didn't initially see it. So there seems to be a similar sex difference. So we do think that there's some biology behind this that might be common in mammals that we'd like to learn more about. And we have published a review, again, of clinical and animal data in 2021 on this. So how do these deficits arise? Where do we get them from? Well, there's pillars or hallmarks of aging. You can read about these. They've gone up to 15 or something, 14 I think now. There are things like macromolecular damage, like the DNA damage we get from exposure to the sun. There are things like changes in our epigenome. There are other things like there's metabolic dysregulation. Our stem cells stop being able to replace things. And then there's chronic inflammation. And I draw your attention to that because that's the one that we've looked at the most often. So chronic inflammation. Inflammation is a good thing, as I'm sure you know, when it comes in to fight an infection. So it comes in, it does its job, and it dissipates. But chronic inflammation, something called inflammaging, is thought to be a key mechanism that contributes to tissue damage in older adults, right? So we've looked at the role of chronic inflammation by measuring serum cytokines. And cytokines are proteins that are chemical messengers in your immune system. And you can get pro-inflammatory cytokines that promote inflammation. So if they're chronically elevated, they promote a lot of inflammation. And you can get anti-inflammatory cytokines as well. So we'll talk about those in a sec. So with Alice Kane, I'm trying to mention everyone as I go, but she was a postdoc in my lab who's from Australia originally and now has her own job in Seattle, which is very cool. So she and I worked on this project and we measured cytokines. This is IL-6, interleukin 6, in the top panels. And you can see if we plot interleukin 6 levels as a function of how frail the mouse is, there's a nice linear correlation between the frailty score and how much inflammation they had going on. Interleukin 6 is a pro-inflammatory cytokine. IL-12P40 is another pro-inflammatory cytokine. That one appeared to be more closely related to frailty in males. And we see, we've measured 23 of these, and we see different patterns in males and females. But many of the cytokines are related to the degree of frailty. So that suggests that the mice with the highest frailty scores do have the highest level of inflammation. And so why is that, going back to the heart, why is that a problem in the heart? So if you start to get inflammation in the heart, you start to get damage to the cardiomyocytes, you get scar tissue, fibrosis tissue forming, collagen accumulation, and that starts to affect the ability of the heart to function as a pump because it becomes stiff and it doesn't function so well. So we wondered whether chronic inflammation in frail animals that we'd seen caused the heart to adversely remodel or change in ways that are not helpful. And does this set the stage then for cardiovascular diseases? So we measured, again with Alice Kane, we measured hydroxyproline, which is a major component of collagen. So this is hydroxyproline levels, and you can see MA is middle-aged, so this is middle-aged M and F, male and female mice, and old male and female mice. And a couple things here. One of them is that the hydroxyproline, the collagen, was higher in the females earlier on, so it was high in females at all the ages we looked at. It increased with age in males, and so we thought that was interesting. And collagen was significantly higher in the younger females than in the males. So we then thought, okay, let's look at the relationship between this collagen that we measured and the frailty of the mice. And so we predicted that the mice with the highest frailty scores would have the most collagen. And that worked beautifully in the males, but we didn't see any relationship in the females. This fascinating pattern, I wish I had a better answer for you, is something we see again and again, where a lot of things are graded by how frail the males are and not in the females. So there's clearly quite a few things that we still need to understand here. But we did see that the males with the highest levels of collagen had the highest levels of frailty. And sorry, that was measured in the ventricles. I don't know if I mentioned that, but we also did something called E over A ratio with an echocardiography, which is like an ultrasound of the heart. So, you can just put a probe on the chest and measure heart function, do it in people or animals. And we found, I won't go into a lot of the details here because it's not important, but we found abnormal E over A ratios, which all you need to know is that they indicate a stiff heart that doesn't pump properly. And again, we saw the E over A ratios decreased with age in the males, but not the females. And again, we saw that there was this nice gradation of this E over A ratios with frailty scores in the males, but not the females. So it suggests that, at least in male animals, high levels of frailty are associated with this collagen accumulation that is impairing the ability of the heart to function as a good pump. With Rob Rose and Haley Jansen, they are at the University of Calgary. Rob used to be at Dalhousie maybe 10 years ago, still a good friend and collaborator. They actually used some of the same mice and were looking at collagen in the atria, and they found the same pattern in the atria. So collagen was accumulating in the atria as well as the ventricles. When collagen accumulates in the atria, it does bad things to the ability of the atria to conduct impulses and messes up things like that, and this paper also has shown that. So what we think this means is, in the heart, you can get fibrosis in both the atria and in the ventricles. And the fibrosis in the atria can lead to diseases like atrial fibrillation, which is a disease where the atria just almost quiver. They don't contract functionally. And in the ventricles, we think that ventricular fibrosis can lead to a stiff heart and lead to something called HFpEF, which is heart failure with preserved ejection fraction, which is a type of heart failure for which we really don't have very many good treatments. And it's becoming a bit of an epidemic because it accounts now for more than 50% of heart failure cases. So we think that the basic idea is you get microscopic damage from all those pillars of aging, right? Things like stem cells not working anymore, and inflammation, et cetera. And that starts to scale up to affect the organ, right? The atria, the ventricle. And then that starts to scale up to affect the system, and it can lead to system failure and frailty. So that's sort of the basic thinking here. So we wondered if there were other adverse changes in the heart that are associated with aging and frailty, pardon me. We looked at calcium channels, which are a type of channel that lets calcium into the ventricular cells. We also looked at the myofilament proteins. I've done a lot of work on this. I'm just sort of, presenting a few snippets, but this is calcium channel CAB 1.2, which is the voltage-gated calcium channel, which you may have heard about in your studies. And we even found the protein expression, as well as the ability of the cells to contract, decreased as frailty went up. This was done with Hyrad Faraduni, a former PhD student in my lab. We also, with Glenn Pyle, who's here at Guelph, we've collaborated for many years on myofilaments, and he's helped with assays on some of the tissues we've sent. We found that desmin phosphorylation, so desmin's a contractile protein. When it gets phosphorylated, it forms toxic protein aggregates in the heart, and it's seen in heart failure. It's also seen in mice with high levels of frailty. So frailty is associated, but interestingly, only in males again, and all the myofilament changes we saw were only in the males, not the females. So it's a fascinating and reproducible pattern that, again, as I said, we don't understand yet. But suffice to say that there are other microscopic problems that are happening, changes in desmin phosphorylation, changes in the calcium channels that are impairing contraction, forming toxic aggregates, and reducing the ability of the heart to contract. And this could predispose towards HFrEF, or heart failure with reduced ejection fraction, more conventional type of heart failure. But again, the idea that there's these small things that are happening that start to add up and add up and add up and lead to system failure and frailty. So why is this important? Well, because maybe we can do something about it, right? Like, that's the exciting part for me. We know, this is from our Nature Aging paper, but reviews all kinds of data which show that things like a balanced diet, calorie restriction, which sounds sad, but anyway, high levels of education, new class of drugs called geroprotectors, wealth, safe neighborhood, social engagement, these things tip the balance towards fitness, right? And altogether, they actually promote a fit older person. High-fat diet, exposure to radiation therapy, poor maternal health, low personal wealth, polypharmacy, taking like tons of medication at the same time like many older people do, low childhood education, stressful environment, and low social position. Some of these things are modifiable, some of them are not, but they predispose towards frailty. And so in animal models, we can actually start trying to look at some of these things and see whether or not we can actually try to add things that promote fitness or reduce things that promote frailty. And can we actually reduce frailty, which might ultimately reduce all this damage that's happening in the heart? So one of the things that I've worked with, and again, I encourage you to think about a career in science. This is a group in Australia. I've been to Sydney a few times, and I don't mean Cape Breton, which I've been to as well. It's very nice. But I've been to Sydney to collaborate with these people at the University of Sydney on this polypharmacy diet. So they've actually got a model where they're feeding mice 5 different drugs with a high drug burden index, like lots of sedative side effects and anticholinergic side effects, if you know what that is. So these drugs have many adverse effects, right? And so what they've done is measured frailty in these mice. So they're older mice, so they've exposed from the age of 12 to 21 months to this high polypharmacy diet. And their frailty score here is plotted against age. So I'm going to take you through this. So this is the control in black. So you can see that's got the lowest frailty score, and it increases with age. The 0 dBi, like the low drug burden index, maybe acetaminophen, things that are not all that challenging, really didn't do much to frailty. That's the 0 dBi. The low dBi, again, didn't do that much. The high dBi significantly increased frailty. And what was really interesting is when they deprescribed, so they took the mice off these drugs for 3 months, from 21 to 24 months, they were able to reduce frailty. So it shows that it is possible, to drive frailty up and also to bring it down by getting rid of the thing that was causing this to happen in the first place. So that's, again, harder experiment to do on people, but there is interest in geriatric community to try and deprescribe drugs, because many people are taking drugs to treat adverse effects of, and it goes like that. So there's a lot of interest in this idea, and this is an interesting mouse model to test it. We've looked at things that can treat frailty, and so we've looked at the renin-angiotensin system, and the renin-angiotensin system is a system that controls many things, including blood pressure, desire for like thirst, things like that. And when it gets activated, such as in heart failure, you get lots of angiotensin II produced, and lots of angiotensin II can act on receptors to produce things like inflammation. And there's an enzyme called an angiotensin converting enzyme, and you can use inhibitors of this enzyme to reduce angiotensin II. You don't make as much, and you can reduce inflammation. And so we've used a drug called enalapril, that's an ACE inhibitor commonly used to treat hypertension and heart failure. And we fed it to our mice in chow. And they started at 16 months of age. And the females, you can see them labeled here, and the males worked with Caitlin Keller, who was a master's student in my lab, on this project. And so we looked at what happened with enalapril. You can see here quite clearly that the females became frail at earlier ages and much more frail than the males. That showed quite nicely. And we actually stopped the trial at about 21 months in the females because we had a really good effect. But the males took much longer to demonstrate a beneficial effect of enalapril. So you can see here that in the presence of enalapril, they stopped getting frailer, and in fact, it might have even reduced frailty slightly. The males, there was a small but significant effect as well when we treated them with enalapril. Now, this might be hard to see, but what we did was we measured all the markers of inflammation, those cytokines, the pro and anti-inflammatory cytokines, and we looked at what happened in the females. And what happened in the females was 3 potent pro-inflammatory cytokines were reduced by enalapril. So it was actually reducing inflammation in the females. In the males, it had no effect on pro-inflammatory cytokines. It actually made more of this anti-inflammatory cytokine called IL-10. So same drug, more effective in the females. It was damping down inflammation. It was promoting this anti-inflammatory cytokines in the males. So completely different impact on inflammation, but beneficial effect in both. So it's important then to consider things like age, sex, and frailty when you're trying to design these type of studies, because if you only were to use one sex, you would have concluded, oh, it's all by an anti-inflammatory cytokine. The last thing I want to say, and it looks like the time will be good, we've started to try and do exercise. Now, exercise is known clinically. It's the pill we all want to take, right? It's the, take the exercise pill, and then we don't have to go to the gym, right? But I personally, believe in this stuff and do go to the gym, trying really hard. We wanted to put the mice on an exercise program. We used males and females, which hadn't been done. And we looked at the impact of voluntary wheel running on male and female frailty indices. We started them at about 23 months of age. So this is like putting a 70-year-old on a running program after they'd been a bit of a couch potato for their entire life. Fun fact, some of these mice ran a marathon in a period of weeks. And that's like on those little legs, right? And they're older. Some of our favorites actually spent a lot of time trying to figure out how to stop the stupid wheel from moving so they could build a nest in it, right? But it didn't matter how far they ran. The mice with the wheel, presumably because, I'd like to have a Fitbit for them, but presumably because they were, building nests and doing things, they were much less frail than the mice who were sedentary for that same time. And what's exciting about this trial, again, you can see what happened in the males and the females. So this isn't as long as the last one, so the frailty is going up quite slowly, but you can see in the males, exercise, the opportunity to exercise, sort of damped down the increase in frailty that you see over a 12-week time frame. So this was mice going from 23 to, for up to 3 months afterwards. So these were really quite old animals, right? And the females here, their frailty actually declined. So they were able to not just stop it from getting worse, but actually improve it. So far our work is suggesting that females can benefit tremendously, from these interventions like exercise, like something to damp down inflammation. And there's also benefits in males, but we may have to try and find a better combination there. So we're trying to do, this is work done with Elise Bissat, who's a current student in my lab, PhD student, and Scott Grandy, who is in health and human performance at Dalhousie. So we're now trying to put together a lifestyle intervention which allows exercise plus nutritional supplements, etc. We're doing this kind of work in mice and hope to start doing some of this stuff in people. So this brings me to the geroscience hypothesis. And there's a lot, I go to a lot of meetings in the aging area, and there's a lot of excitement now, and there's a whole ton of new drugs that are geroprotectors that are in trials. There's senolytic drugs, if you've heard of them. And the idea is simple. So if frailty arises because you have all of this damage happening at the cellular and subcellular level through things like inflammation and stem cell senescence, you start to get impacts on organs, not just the heart, right? But the pancreas, the lungs, the brain, the kidneys. So multiple systems are going to be dysregulated. So even though medicine is designed to sort of look at, I'm a cardiologist, look at the heart, I'm a nephrologist, look at the kidney, a lot of these things are happening in older people at the same time. Like many systems are going at once because they get this damage accumulating. Leads to multiple organ dysfunction. So not just the heart, but other systems can be impaired. This then tends to lead to system failure and frailty. So the geroscience hypothesis says if we treat these fundamental aging and frailty mechanisms, we can slow this decline in organ function. You're never going to prevent it, but you can damp it down and delay the onset of these diseases of late life. And ultimately, improve lifespan, but lifespan is not all that interesting if you're really unhealthy. Nobody really wants to live, for a really long time in a state of incredibly poor health. But people want health span, right? They want to improve their health span. And so that's the type of work that I'm interested to, do. So just to conclude, I hope I've convinced you that age-associated changes in sex steroid hormones can affect the heart, and this can help us modify some treatment strategies like cardioplegic solutions during surgery in older animals. It could help us identify other targets as well. It's not just age, but it's the degree of frailty that seems to, set the stage for chronic diseases, cardiovascular diseases in late life. I think the effects of frailty on the heart are sex-specific, and they're probably easier to understand for males than females. I think most models of frailty can be used as really interesting mice to test geroprotective interventions and promote healthy aging so that people actually can, quote unquote, die healthier. In our study with exercise, the mice that were offered the opportunity to exercise, there was no difference in mortality, but they died healthier because they were less frail when they died. So it's an interesting idea and one that is gaining a lot of traction in clinical medicine. And so I would like to thank CIHR and the Heart and Stroke Foundation. I mentioned pretty well everyone I think on this list as we were going along. Also Peter Nickel, a longtime technician in my lab, and Jake Wanju, who helped with a lot of this work. And I thank you for your attention. Oh, yeah, well, okay, thanks. Okay, any questions? I don't, do I have some time, right? Yeah. Okay. Yeah, sure. Oh, cataracts, you're asking? Okay, so your question, if I'm hearing correctly, is that you're wondering about the frailty index because one of the items is cataracts, right? So you can create a frailty index from any number of different things that are bad for you, like test abnormalities, clinical deficits. So cataracts could be there as well as some of the biochemistry. The better frailty index is probably the bigger one. So you can actually fold the lab frailty values in with the clinical frailty values if you want, right? So cataracts are one thing that could be bad for you. So it has to be something that's age associated, and it's bad for you because you can't see. And in mice, that's a problem because out in the wild you get eaten by something, but you'd never make it that old, right? So cataracts are there. Tail stiffening is one of the items, but that actually reflects a degree of collagen cross-linking So again, it's going to manifest in multiple tissues and organs, right? So not just, it's an indicator of that going wrong. Swollen abdomen could be obesity. It could also be a tumor. It could also be ascites from heart failure. So, things like that. So a lot of these deficits, may reflect underlying pathologies. And everything we have in the index, we actually have looked in the literature and validated that, it occurs with age in this model of mice, and it is bad for you in some way, right? Like patchy fur loss, alopecia, it's not just like thinning at the temples, right? But it's actually like they're losing fur, and that's associated with inflammation in C57 black 6 mice. So, it's a good question, but that's how we've justified all the items that are in there. So we observed them, and then we went and did research into the literature into how these things are linked to aging in mice. Does that help?
Yeah, because the thing is, I made that after the frailty index reaches a certain value,
Yeah.
Yeah. So I was like, if there is a cataract, so if there is a cataract, it's that one more value in the frailty index. And I think if the cataract maybe, longer.
But maybe they weren't that, yeah, so somebody with a cataract could live a really long time. It's not just any one item, right? That, like most of the time, if you broke your finger, you'd be fine, right? But the idea just is if people are kind of up there with a frailty score around 0.5 or 0.6, they just have nothing, like they don't have any reserve. So you think about it, what's happening in the body, right, is you get damage, right? We get damage like, sun damage, all sorts of things. You get damage happening all the time. But then we also have repair mechanisms, right? So you're in a kind of equilibrium between damage and repair. And so people who become frail are starting to accumulate, they have like higher damage rates and lower repair rates. And in fact, we actually modeled that and published it in 2022 with a bunch of physicists. Actually, Andrew Rutenberg, who's here, if you love this stuff, he's here at the, he's from Dalhousie, he's here today in the physics department giving a talk at 4. Interestingly, I ran into him, it's just like, whoa, what are you doing here? Anyway, so yeah, there's 2 of us here from Dalhousie today. But he's talking about the physics, and so you can model these processes, right? And you can look at how things like exercise, can affect both the rate of damage, like it can reduce the rate of damage and it can increase the rate of repair. So these are driving these processes in some way, right? And so if you've got people who have like accumulated so much damage and they have very little capacity for repair, they really can't tolerate another deficit. So it isn't so much about any one thing. The idea just is if you become really, really frail, even small things like a small medication error, it could be fatal for a frail older adult because they just, where somebody younger could bounce back, somebody older just doesn't have that reserve or whatever you want to call it, resilience, Is that good? Really good question. Yeah. Yeah.
Because frailty tends to be applied to younger as well, like younger with health issues, they're not the same age.
Sure. The question is, can the frailty index be applied to younger people with health issues? Frailty, they have measured it in some of the data sets as early as age 15, but it is going to be really, really low on average. But in the individual, it could be higher, right? So people with some of the, with a chronic disease that starts early, right, could end up with more damage early on, right? So that becomes a sort of hit that's harder to repair from. So this is not something that's been studied a lot, but you think about, people who were born prematurely, for example, like they have a lot to overcome so they could be frail, but then they could improve a lot. So you might see their frailty going way down, right? As they develop. So it is a concept that could be applied across the life course for sure, yeah. Yeah?
Hi, I know you also talked about the genetic changes.
Yeah.
What is that relationship to cardiovascular disease?
Oh, wow. A relationship with epigenetic, I haven't done any work on that. So, you could end up with, because the mice that we see, right, have so much difference, I think that they must have epigenetic changes going on to make them so heterogeneous, even though they're supposed to be, they're all eating the same thing, they're all in the same cages, they're often litter mates, and they're so different. So I actually think that those type of epigenetic changes are probably key and part of what's going on in terms of their frailty levels, right, and the heterogeneity and frailty. How it relates to the heart in particular and frailty, I can't really answer, but yeah, interesting idea. Yeah. Yeah, okay, the question is about, is there anything I can say about the morbidity and mortality idea? Yeah, there's a person that I know really well called Dr. Ruth Hubbard, who's in Queensland University in Australia. And she is working a lot on that, and we're trying to figure it out. Like, people have grants going in on that question. Like, what is this morbidity mortality paradox? She's a clinical geriatrician who does a lot of research on people, right? And so she studied factors that could predispose towards higher frailty scores in women. So part of it could be gender, like in terms of women might, as a group, be more inclined to reveal more things to a clinician about their health than men, right? So there could be gender roles that play a role, and people have speculated on that. Not me, I haven't written on this specifically. I've been more interested in the fact that we can see it in mice suggests that there's also some underlying biology. But it's probably multifactorial, right? It could be, in addition to things like that, it could be that women have children. And so she's looked at parity, you know, how many babies you've had. And her research has suggested that around 2 kids is a sweet spot, and more or less is associated with higher frailty, which I think is really interesting. She's looked at immune differences in males and females and suggests that they may be responsible. I mean, I saw some pretty interesting differences in inflammation between the sexes, so I think that could also play a role in people. So I think it's probably multifactorial, right? Yeah, and there's at least one person I know putting a grant in to try and figure that out in mice, and it's not me. Yeah, I mean, I'm still doing some work on that, but it's not a major focus. Necessarily. I'm kind of more interested in the intervention side right now, and there's tons of work to do here, right? So there's, yeah, but interesting. Yeah. Oh, thank you. Okay, so the question, as I hear it, I'm repeating it so everyone can hear it, and also so I got it correctly. You want to know where I see this research going next? Yeah. Okay, so a lot of people have used this tool. Like, I've been invited to speak a lot over the last 10 years, all over the world, about this, because people are really excited, because nobody really thought about trying to measure frailty in animals, right? And so what people are using it for now, a lot of them, is to do trials in animal models that they only used to measure lifespan in any of these studies, right? And as I said, that's not sufficient. Just looking at making a mouse live longer, it doesn't speak to whether or not they're living healthier, right? So people have seized on this as a way to kind of get a handle on not just lifespan, but also health span. So people are using this for all kinds of different intervention studies. And then with the idea that if we can find some good combinations, that we could then pivot that to work in people. So that's a big way in which the scientific community in the aging field or space as well, especially, is using this approach to try to understand how we can make people age better and healthy for longer, right? So that we compress late life morbidity. So we have a very small time in late life where we're feeling really sick, as opposed to sort of spending 10 years with really ill health, which is not what people want. So I think that's kind of the way in which people are trying to use it. Personally, but I'm interested in that aspect as well. And I'd like to try to find interventions that are really easy to translate clinically, used in mice, that we can then say, okay, now we're going to try a clinical study in people. And so that's what I'd like to do. Thanks for the question.
One more question over here.
Okay, just,
Yeah.
Hi, how are you?
Good.
Thank you so much. I wanted to quickly ask you about one of the pillars of aging, specifically childhood education, and how you think that impacts frailty, perhaps.
Right, so education is protective against many late life diseases. It's protective against dementia. And early childhood, poor early childhood education has actually been shown by some really interesting work by a number of people. The studies are talked about in our Nature Aging paper, but has been shown to be a profound, it sets people up for a higher level of frailty later in life at an early age, and it's a modifiable risk factor, right? So it's sort of tempting to say, look, we really need to do something about that. right? So an educated population is going to be on average, again, individual different, but on average, so these are all average things, will be healthier. And that would start early in life. Like some of the stressors that you saw, like poor maternal health, that's impacting on the offspring, right? And so that's like setting somebody up with epigenetic changes that could be, profound later in life and manifest as different diseases, right? So the way to not be frail in late life is to try to arrive at about age 50 or so with as few deficits as you can manage. So exercise is the thing that I kind of go rah-rah about, because it's the one thing we do know that does work. And, but it would be nice to try and actually say exercise plus, and we could get a better outcome, right?
You guys stay there for 1 sec. I need the microphone.
Which one?
The one that is just amplifying your voice. Yeah, that one.
Okay.
Yeah, we don't have to. Okay, one more thanks for